We're good. Got a few more minutes.
No, the when you get out of the VR, it's a really weird feeling. It's like you just woke up from a nap or something because your surroundings are completely different than they were just like 1 second ago. I don't know. I feel like I'm like, whoa, all at once. In our experiment, we're testing to see if our conscious perspective of the environment we're in changes our responses during exercise. Particularly, we're challenging if there's a mismatch from what you see in your environment compared to what the temperature in your environment is in reality. We're using virtual reality technology in order to achieve this, and so our participants might be in a world where they see snow or nighttime, and in reality it's 90 degrees in the chamber, and they could also have a hot dry desert and it's room temperature.
I feel like I know the path of your GI tract now.
It's exact.
Getting to know each other there.
Our results could provide an insight into how our conscious perspective influences our thermoregulatory controls. This could be important as we incorporate more and more technology into our daily life and we adapt to our warming world.
Hello everyone, this is Dina. I'm a second year graduate student majoring in Human Physiology. I'm currently doing my research in Node Lab, and my study investigates the relationship between lower limb muscle activation and cortical activation during working in two groups of people, the individuals with knee osteoarthritis and healthy controls. I'm utilizing the combination of SEMG to assess muscle activity and FNIRS to assess the brain activity.
Hello, my name is Chen, and my project involves using human physiochemistry method, which is a labeling technique to study the brain. And specifically, I'm studying thalamus, which is referred to as relay station in the brain. And in thalamus, I focus on one first-order nuclei, which is called LGN, and one higher-order nucleus, which is called medial dorsal. And I'm really interested in finding out the ratio between the synapses, especially the excitatory versus the inhibitory synapses, because we know that the ER ratio is very important to keep the brain normal functioning. And given that the part of data that I'm collecting is missing in the literature, so I think there is a significance I'm getting in my work.
Hi, I'm Rishi Bhomik. My research is also studying inhibitory neurons, specifically inhibitory interneurons in the brain. My region of interest is the hippocampus, and I'll be using 4 protein markers: relin, vasointerstitial peptides, somatostatin, and cholecystokinin in rat hippocampus brains, and whichever ones will stain and work the best using immunohistochemistry, like Chen said, those will be done in monkey and or human brains to identify subsets of inhibitory neurons in the hippocampus, which have not been done with these markers previously. And both of our experiments will be using the confocal microscope right behind us.
Hey everyone, my name is Evan Nessen. I'm a second year graduate student at Boston University in the Master of Science program in Human Physiology. I work in the Queer Lab, which is led by Dr. Maria Bouchard and Dr. Lisa Roberts. My thesis is investigating psychosocial influences of blood pressure and physical activity status among queer and heterosexual young adults. There's some research that indicates queer individuals are at higher risk of cardiovascular disease. So we're taking real-time objective cardiovascular measurements and seeing if there are any differences between queer and heterosexual young adults, and if there are any factors that either ameliorate or make blood pressure worse.